Okay, hello and welcome. Thank you for giving me this opportunity. I'm Aritra Chatterjee. I'm a faculty at IIT Guwahati in India. And the work which I'm mostly interested in is to actually see the role of initial geometry and substrate mechanics in collective cell migration. So why collective cell migration is important? It's an important feature of morphogenesis and tissue homeostasis. And while there are some experimental work that says how geometric or mechanical cues can affect this, these things are underexplored. In the lab where I did my PhD, I specifically want to focus on a particular work which is of interest to me and to model, where you can see we are studying this collective cell migration, where the initial confinement geometry is different. They have very different curvatures. So when we actually try to see how these structures and these cells move when the initial confinement is lifted up, we see that there is very high curvature sensitivity in the sense that for circle uniform curvature, it moves radially outwards. If you see this push-form structure in regions of a very sharp curvature, it doesn't move. It only moves sideways. So there is this mechanism where cells are able to sense these curvature distributions, and the relative position within this confinement is influencing its motility. So as this is further confirmed by these vector maps, where we could actually quantify these measurements, where depending on how the initial cells are, their movements were very different from whether they were at the vertex, where there's a sharp change in curvature, or they're at the sides. For the circle, it is uniform curvature, you see everything moves as it is, but for the square or the cruciform, where we have a differential change in curvature spatially, you see the cell motility is different, the later cell emergence is different. So this is something we actually, I would like to study and model in CompuCell. And then also it was found out by my ex-lab mates who worked on this, is that the actomyosin contractility plays a very important role. So two different cell lines were done where we have tried with MDCK, where actomyosin contractility is higher than MCFs. You see that the velocity fields are higher, as well as when you do these experiments where you treat these cells with blebistatin, which inhibits myosin, you see that the migration patterns are much, much less as compared to cells. And just if possible, a future extension that I'm really interested in is to actually also couple this with the underlying substrate mechanics. There's a very recent study from the group of Aritra Chatterjee where they have cultured these cells on hydrogels that have similar elastic modulus but different relaxation time. So faster relaxation time would be to say it's more viscoelastic, the loss modulus is higher than the other one. And the cells are able to sense this difference in viscoelastic parameters, and it is characterized by higher contractility and less speeds. So this is so how the cells are sensing this change in the substrate, how cell-to-cell adhesion and cell matrix adhesion is changing, something we want to understand. So this is the modeling goal part. So we want to understand the mechanism by which cells can sense this geometric confinement, as well as understand the mechanical cues. And the hypothesis is that the cell motility is sensitive to the local curvature, as well as it is the cell substrate interactions depend on the mechanics. So how we are trying to predict this? Basically, we want to look into these different mechanisms where it should incorporate from the cell-to-cell adhesion, cell matrix adhesion, and cellular contractility, and whether these parameters are actually sensitive. So whether it is an outcome, the simulation where we just put the confining geometry and same parameters for all cell and we see these results, but we have to tune them is something I want to understand. Why CPM? It's lattice-based modeling, which has previously been implemented. The Hamiltonian approach helps us to capture this net energy as three different terms, where I have a active energy term, which takes into account the leader cell motility, there is cytoskeletal modeling, passive energy, and cell-to-cell cell matrix addition. I was very much impressed by yesterday's Querying Brad's talk, which kind of gave me a lot of insights on how to do this, as well as some previous paper from Amit Pathal's group, where I have taken these parts where people have tried to model this using these different steps. The ideal inputs would be the initial geometry, energy inputs for these three different active, passive, and contact terms. And then we will try to see whether this actually I have read, because it's something I'm not sure of. I'll appreciate feedbacks. Whether the cell matrix interaction energies should be different based on the cell-to-cell layers or something else. And the outputs, the model should be able to capture the migration patterns, the velocity fields as a metric of whatever inputs being give. That is what we try to do. So this is the project plan. Ideally, I thought of that there should be 3 different cell types: the medium as it is, the cells, and the ECM. The ECM we kept frozen, and the initial geometry can be customized based on the PIF files. As Chandra showed yesterday, there should be the contact plugin, which should take into account the cell-to-cell and cell matrix interaction. And the volume and surface flex can be used to actually take into account the cytoskeletal remodeling. And as yesterday query in Brad showed, I need to figure out how this external potential plugin can be used to find out the reaction process. These are some of the resources on papers which is already there. I have uploaded the slides, so you can find it. I am actually from a mechanics background. My research interest in cell tissue mechanics of soft matter. I do primarily experiments as well as some constitutive modeling, which is in continuum mechanics, and some MATLAB programming. I'm new to CompuCell3D. When I was working as a postdoc at Purdue, I first heard of this as well as part of Embryo. And I attended 1 or 2 lectures, but I really appreciate some teammates who have prior experience with Python programming like this so that we can tackle these problems together. Also very much open to any long-term collaborations along this field where I can help. Thank you.
I was going to say, since Maciek is on the call today, this is a case where having Roland's ability to couple directly to a finite element solver for the viscoelastic material might be really interesting. To begin with, we could do something like what Karine did or what Tien did, where you have a mesh of virtual cells which are triangulated to represent the compressible substrate under the cells. And then you can use links between the cells and the substrate. But the spatial resolution of that will be a little bit limited compared to that what you could get with the methods that Roland developed. But I think you'll be certainly for the hackathon, this is a great place to start.
Thank you. Yeah, I really look forward to learning that and finding it.
I'm interested in bone remodeling. So the functional unit of bone is the osteon, which is this column. Basically, you can't really see it because this is a top-down image, but it's a column of bone matrix and bone cells. There are a few different types of cells involved in bone remodeling. So you have osteoclasts, which remove bone matrix, osteoblasts, which synthesize bone matrix, and then osteocytes, where the osteoblasts get trapped in the matrix and they just become part of the bone network. There are the cell speeds have been discovered under electrical gradients in a medium, and so they move at the rate of micrometers per hour, so it's very slow. And these cells interact with teams to remove and then synthesize bone matrix as they move through the osteon, which is what I'm interested in exploring. I'd like to see how the leading front of osteoclasts actually work, how these cells move through the osteon. Is it like a plane? Or like a disc? Is there a convex or a concave shape? Then I would want to understand how these disorders in these in the mechanics of these cells lead to diseases like osteoporosis and Paget's disease, which are triggered by this imbalance between osteoblasts and osteoclasts. So I'd be interested in modeling the basic multicellular unit of this process and modeling how the osteoblasts and osteoclasts interact, predict the microstructure and of the remodeling process, and then to be able to adjust the parameters that would lead to reproducing the microstructure, the defective microstructure in pathological tissue. Like, for example, making the volume of osteoclasts, osteoblasts, that is, less than the osteoclasts. So you might see something like the bone getting progressively removed, and then the defects, and maybe that would resemble the defects that you observe in those diseases. So I think that this is probably pretty feasible to do. I think at least I think there's something that is feasible within a weekend, because I think that the cells that are involved are pretty small. I don't think it's like a huge amount of cells involved in these remodeling teams. The cell types, there's not many of them. It's mostly just the two that I mentioned. The ECM is static, so it would be simple to model, but it would be difficult, I think, to model something like stress, mechanical stress on the bone, unless you use something like continuum mechanics. The long time scales involved are also a concern, because bone remodeling takes a very long time. And the signaling networks involved, they might be very complicated. I'm not really sure. Here's some papers that I cited. And then finally, teammates. I'm looking for pretty much anybody. I don't have a big background in biology, especially cell simulation. I'm mostly familiar with bioinformatics, genomics, things like that. So pretty much anybody who would want to help would be welcome to help. And then if there's some potential here that goes beyond the hackathon, then doing a research paper might also be interesting. And so that's pretty much it.
That's great. I would encourage you to look at the published papers of David Basanta, who has studied bone remodeling primarily in the context of cancer metastasis. He studies cancer metastasis of prostate cancer to bone, but he has agent-based models of bone remodeling with pretty detailed subcellular interaction networks. And we've talked about trying to re-implement some of his models. He has his own framework for doing it. We've talked about re-implementing some of his models, and probably that would be not a bad place to pull some information and some ideas about how to represent this.
All right. Well, thank you, Nathaniel.